I'll make all those cancer treatment top hat things as homework. We're going to start the next bit about meiosis and genetics, but we're going to first go over some of the stuff we already learned. So, the cells in your body that do give rise to sperm and eggs are called what? Basically happen. Okay, so if we look on the bottom here, there's three answers for three of these different questions: enzymal, anchorage dependence, cell cycle control, contact inhibition. Cancer cells differ from normal cells because they divide when they shouldn't, and that's loss of what? Invading, loss of what allows invading surrounding tissue, contact inhibition, or anchorage dependence? What do you think? Contact inhibition or anchorage dependence? Huh? Yeah. And then obviously metastatic cancer, loss of anchorage dependence. Remember, I talked about radiation therapy. It damages the cells so that the tumor suppressor system actually recognizes damaged cells and causes those cells to undergo apoptosis. So, which of these? Radiation therapy is least effective against contact inhibition, anchorage dependence, in other words, invasive cancer, metastatic cancer, or causing cells to kill themselves. Anybody? Yeah. Why is it good to take multiple biopsies from a single tumor? Because unregulated cell division leads to increased genetic diversity. Remember, cells are dividing every once in a while and new mutation starts. Yeah. Because a given drug may be effective against only one part of a tumor with one kind of mutation. Yeah. Because tumor evolution can occur, obviously it is. Keep dividing, the more likely it is they're going to develop unregulated cell growth because of mutations, or it should be D. All of these are true. That. Okay. Okay, so here's the cell cycle. Remember, G1, most cells are in G1 98% of their lives. Some, all of their lives, like neurons. Once they're there, they don't continually replicate. Whereas other cells, like skin cells, are more likely to go into the rest of interphase. If a cell is going to divide and replicate, it needs a signal first, which it gets in G1, that institutes or causes the synthesis of all the DNA. So there's now two full copies of the genome in this cell. cells need energy to replicate. You're not just replicating the DNA, but all the other things in the cell, including mitochondria, translational machinery, all kinds of different things. So a cell has to grow enough and have enough nutrients so that it can actually undergo replication successfully. Otherwise, it would start to replicate and then it wouldn't make it because it wouldn't have enough resources. And then finally, the actual replication into two different cells starts with mitosis. And there's 4 major phases. Prophase, where the chromosomes or the DNA condenses in the chromosomes. Metaphase, where all these chromosomes, they're not duplicated, line up along the middle of the cell. There are these microtubules that come down and connect to these chromosomes and then start pulling apart so that each cell has a full complement of all the chromosomes. And the pulling apart phase is called anaphase. And then nuclear envelope starts to reform. So in prophase, the nuclear envelope starts to break down, which is where all the DNA is, and that condenses. And then telophase is where it reforms, and then cytokinesis is finally where you have 2 daughter cells that are identical to each other unless there's any sort of mutations. CP. Checkpoint, and then MCP. Okay, so there are three checkpoints. One in G1, after a cell has to have the right signal to replicate. If it starts replicating without that signal, the tumor suppressor system kicks in. G2 the cell has to be large enough. All the DNA has to be double the amount. And then in mitosis, the chromosomes have to separate equally. So each side gets homologous chromosomes instead of one getting 1 chromosome and the other getting 3. Okay, we'll stop there. And okay, so this is where we left off. Okay, that's probably better. So a lot of organisms reproduce asexually. On the top left is a bacteria splitting into 2. I'm not sure what it is top right. Bottom left are 2 single-celled intestinal parasites called Giardia, and they're splitting in 2. And then the planaria on the right, they're actually multicellular, but you can actually chop them up and they'll completely regrow themselves. So all these things do it by way of mitosis. Okay? mitosis. That means cell replication. Same thing as when I'm, let's say, replacing my red blood cells. Red blood stem cells in my bone marrow, by mitosis, make daughter copies of themselves, and then those mature into red blood cells. Same sort of thing. So, in this case, daughter cells, or daughter organisms, are identical genetically, 100%. If you have certain alleles, those identical daughter will have the same alleles. Okay. So that's different than sexual reproduction. So the cells of your body that do not give rise to sperm or eggs are called what? Anybody? Starts with a S. It's not reproduction. The cells of your body that do not give rise to sperm or eggs. We talked about cancer. What? No. That's not. It's true, but yeah, exactly. Somatic. Okay. The cells of your body that do give rise to sperm and eggs are called what? Starts with a G. That's what happens, yep. So what produces gametes? What cell lines actually produce gametes? It's not somatic cells. Huh? Germline, we'll say. How's that? Okay, so only germline cells give rise to gametes. So if there's a mutation in the somatic cells, can that be passed on to offspring? No. Okay. You can certainly have mutations in somatic cells. If they're bad mutations, then you might end up with cancer, right? So germline cells then can be, if you have a mutation in germline cells, that can be passed on to your offspring. Okay? Because germline cells produce gametes, sperm and eggs. Or in some cases where there's no differentiation, dysgamete, period. So there's some organisms that are like that. Okay. Now, our somatic cells, what does it mean that they're diploid? So, chromosomes. We have two pairs, right, for each chromosome? Yes. One for mom, one for dad. Okay. So, we are diploid, you and I. Now, our gametes, on the other hand, are haploid. I'll show you how in a second. So, let's take a look at this in greater detail. Oh, pairs of chromosomes, like chromosome 1, are homologous. That means there's two of each, one from mom, one from dad. Okay. The homologous chromosome is this whole thing. A sister chromatid, so each chromosome will have two sister chromatids. Let's say the one in red from mom. That's called a sister chromatid. It has the same exact gene loci as the other one, but at each loci it might be the same allele or different allele. So those are called homologous chromosomes. In the middle of each chromosome, there's a long arm and a short arm is a centromere. And so in homologous chromosomes, the centromeres they connect the chromosome. So this is a karyotype, and it shows the chromosomes. We have 23 pairs of chromosomes. 22 are identical, except have different alleles. And then the 23rd pair you either have an X or a Y. Actually, you have either two X's if you're female, an XY if you're male. Okay? And to display it, we say it's a karyotype. So, remember the cell cycle? Which phase would you actually start seeing chromosomes? G1, S, G2, or prophase of mitosis? No. Prophase. Okay? So it's all spread out before it's time to replicate, and then as it enters mitosis, the chromosomes condense, the DNA condenses into chromosomes. And that's during prophase of mitosis. Okay, so we're diploid. That means we have two of each of the 23 chromosomes, one from mom, one from dad. So we have a total of 46 chromosomes. Okay. Now, gametes, eggs and sperm, they only have one of each chromosome. That means that they're haploid, not diploid. So, one of each chromosome, one of the 23, and the 23rd chromosome is either an X or a Y. Okay? So, if a sperm has an X and fertilizes an egg, what sex do you think it'll be? Yeah, female. If it's a Y, it'll be a male. So it's the male gamete that determines sex. Okay. So remember, these are haploid. Okay, let's go on from there. Okay. So meiosis occurs within the germline, and it reduces the number of chromosomes by half. You go from the germline cells are diploid, but they produce gametes that are haploid. Then, when there's fertilization by a sperm, an egg gets fertilized by a sperm, the diploid number is restored. And a fertilized egg is called a zygote. So a zygote is diploid. It now has 23 pairs or 46 chromosomes like the adult. From then on, as it develops, from a single fertilized egg into a whole human being, it's going to undergo millions and millions and millions and trillions of rounds of mitosis. Where you get identical daughter cells. So it starts like mitosis. You replicate the DNA in the S phase. And there's actually two phases of meiosis 1 and meiosis 2. So let's take a look. First of all, if you remember, we've got G1 phase. That's where most cells spend the majority of their lives. It means they're just growing and maintaining and doing their functions. They get a signal to divide, then they go into S phase. That's where the DNA is synthesized, so you have a complete, one whole another genome. You've now got two genomes instead of one. G2, that's growth and preparation for mitosis. And then finally, it enters prophase, where all the DNA, which is all kind of spread out, condenses into chromosomes, and the whole thing happens. You go through prophase, metaphase, anaphase, and telophase, and then cytokinesis. So this is prophase, where the chromosomes condense, metaphase, and then they separate during anaphase and telophase into two daughter cells. Now, meiosis, this only happens in germline cells. They go through two phases, meiosis 1 and meiosis 2. The first thing that happens is these chromosomes, remember they're duplicated, and they form what are called tetrads, pairs of homologous chromosomes from mother and father. From the individual's mother and father, not the offspring. And they actually, there's something they do, it's called crossing over, which they exchange genetic information. We'll get into that a little bit more later. Then they end up along the metaphase plate. And then finally, the homologous chromosomes end up with sister chromatids remain together. That means after crossing over, you still got homologous chromosomes. But that means there's still one from mom, one from dad, except for some genetic exchange. Okay. And then you do the same thing again. We'll get into this more explicitly, where you then, there's no more chromosomal duplication, but instead of having 6 sister chromatids, each chromatid separates individually, and you end up with 4 gametes. So, like with mitosis, you end up with daughter cells that are identical, Well, here you have individual gametes and each one is haploid. And since there were actually chromosome duplication, that means 4 sister chromatids. Well, those split up into 4 individual chromatids. And that happens during meiosis too. Okay. Let's see here. Okay. You might want to start on the meiosis in Biol. It'll get you familiar with it just like the mitosis did for that as well. Okay. Now, so meiosis occurs in what? Germline or somatic cells? Germline. And they produce haploid or diploid cells. The fertilized egg is called what? Yeah. And it is haploid or diploid? Huh? Haploid. So, the fertilized, oh, sorry. No, my mistake. You're right. Yeah. Never mind. Didn't want to get ahead of myself. Okay. Diploid. Yeah. Okay. So, the gametes are haploid. The zygote, the fertilized egg, is diploid. Okay. Any ideas why there's sex? Anybody? What's that?
To reproduce.
Okay, but why sex? Why not asexual reproduction? What? Okay, we'd all look the same. Okay. So sexual, we create lots of differences. So you have genetic diversity. Now, the real reason that we have sex is because, of course, it feels good, right? That's the only thing that we really care about. But in terms of biology, sexual reproduction results in genetic diversity. Now, there's something kind of interesting. Okay. With asexuals, let's say that each individual can produce one copy of itself. Okay, now with it. Okay, 10 identical offspring. Okay. Which one has higher fitness? Asexual? 10 versus 5. So, why should there be sex when you get twice as many offspring from asexual reproduction than sexual? And yet, sex is ubiquitous. Lots and lots and lots of things do it. So there must be some benefit. Otherwise, selection would favor asexual reproduction. The thing is, with these offspring from sexual reproduction, they're not identical. In fact, they have a lot of genetic diversity because of all the scrambling going on that happened during meiosis. That's why, well, I'll show you. Okay, so remember, you have 23 pairs, and each gamete gets 23 single chromosomes, but which one it gets is random. It could be the one from mom, or it could be the one from dad. And that's what I mean by each one is random. From one cell, we'll get, let's say, one from mom, the other from dad. And that same cell will then get the next chromosome, number 2. It might get the one from dad, and the other one goes to mom, right? So you have all this random assortment going on. So it's called independent assortment since you have 2 of each. The combination that you can have is 2 to the 23. So 2 squared is 4, right? 2 to the 3rd is 8. 2 to the 23 is 8 million types of gametes that are different from one another. And then the female produces these eggs, and there's 8 million assortment, and male 8 million, and so you can end up with 64 trillion genetically different offspring that your parents could potentially produce. So each child is a unique combination of parental traits, except for identical twins. So all that would have had to happen is, instead of the sperm that fertilized the egg that became you, another sperm, and you wouldn't be you anymore. You'd be an entirely different individual. Okay. Monozygotic twins. What happens is you end up, remember, we have an embryo from fertilization, from a sperm and an egg, and then mitosis, it starts dividing. At a certain point, once in a while, an embryo can actually divide in two. And then both of those start dividing, and they end up becoming two identical individuals. Those are monozygotic twins. Now, it doesn't mean they're totally identical, because they have the same genes. They share mostly the same maternal environment inside the womb. But once they're born, they start diverging in terms of what's called their genetic regulatory elements, or epigenetics. So you can actually, that's why you can actually have identical twins that aren't necessarily identical. Years ago, I went to Santa Monica College, and at the espresso bar, there was this Hungarian girl, and every time I went, I usually got a coffee in the morning. I need something for liftoff. And she seemed like she was a downer. She just wasn't very happy. And then some days I need an extra liftoff in the afternoon, so I go back, and same girl, but this time now she's bubbly and happy, and how you doing and all that. So I thought, well, maybe it's just takes her a while to get going. And then one day I go back, and there's two of them. One was sad, one was happy. Even though they have identical genes, I don't know what happened. Okay, but identical twins certainly share all the genes. They have the same alleles. The only difference is that their environments diverge, and our phenotype, which is my anatomy, physiology, behavior, development, all that kind of stuff, that's actually a genotype interacting with the surrounding environment, whether it's a cellular environment or the outside environment. And so once twins are born, they kind of go off on their separate pathways. Many of the same phenotypes they still have. Now obviously they're likely to live in a similar environment, but once in a while twins get separated at birth and go off with different parents. And I'll show you a little video eventually about what that happened and what was kind of interesting. In contrast, dizygotic twins, the female releases 2 eggs instead of 1 during ovulation. Each gets fertilized by a different sperm. So they're just like brothers and brothers, or brothers and sisters, siblings. They're half related to each other. whether it just so happens they're going to be born at the same time, but they're basically like brothers and sisters in terms of who they're related to. Okay, this is a roundworm. It produces sperm. It stores the sperm. It then switches production, and it also produces eggs. And then the eggs can be fertilized by another roundworm sperm and vice versa. Or if there's no other roundworm around, they can actually use their own sperm to fertilize their own eggs. Now, so they're hermaphrodites. That means they can produce male and female gametes. A lot of plants are hermaphrodites, right? They produce, a flower will produce pollen and ovules, okay? Usually, there's mechanisms to stop them from fertilizing themselves, but not always. Still, what they do here, this is sex. The same sorts of things. Meiosis happens, all that kind of stuff. Okay? So, even though it's not the kind of sex that we have, in other words, these guys can have sex with themselves and they're perfectly happy. Okay. So in terms of the cycle of sexual reproduction, you have fertilization, so you have a zygote. Then you have millions of rounds of mitosis as the zygote develops into an embryo and then develops into an embryo with different parts and organs and all that kind of stuff. And then at a certain point, it's sexually mature, and it can then start producing gametes of its own and then fertilize those. So your somatic cells have 2 of each chromosome. Therefore, they are diploid or haploid. Diploid. What are these pairs of chromosomes called? Huh? I heard it right. They're homologous. Yeah. Okay. Okay. So the specialized form of cell division that occurs in your germline is called what? Mitosis or meiosis? Meiosis. And gametes have 1 of each chromosome, and then therefore they're diploid or haploid? Okay. Now, I kind of alluded to this. For now, we're just going to say a genome is like an instruction manual. in terms of the genotype and then the phenotype. And essentially, we get 2 instruction manuals, one from mom, one from dad. It's not really quite like this because, as I mentioned, during development, phenotypes develop, and it depends on the interaction with their environment. It can be a cellular environment, it can be the outside environment, right? So they're not exactly, you can just go through and read it and you'll get a person. It doesn't work that way. Genes are expressed in terms of the environment that they're in. I just want you to understand that. But for now, we're just going to say there's a genetic instruction manual. So here's the hemoglobin gene. Remember, in terms of DNA, we've got 4 letters, A, T, G, and C. This is the whole hemoglobin gene. And it's got a whole bunch of amino acids. These are only some of them. Leucine, tyrosine, prosine, glucine, lysine, serine. And if we actually look, I'll look at this one. Okay. Now, this is the normal allele, HbA. It's the normal hemoglobin allele. Some people have a mutation, and it's called sickle cell mutation. So you've probably heard of sickle cell anemia. It tends to be common in places where there's lots of malaria. Because it turns out that if you have an HbA and an HbS together, those red blood cells are difficult to enter for the malaria parasite. If they're HbA, they're easy to enter. If they're HbS, it means the individual has sickle cell anemia, which is a genetic disease. Okay. So we can actually look at this. So this is what normal hemoglobin looks like, and this is what clumped hemoglobin looks like. Okay. So what's happened here is that the A has been replaced by a T. And so instead of glucine as an amino acid, you now have valine. And it has a different three-dimensional structure. When you have a red blood cell with 2 HbS's, the abnormal, you get something called sickle cell, and it doesn't carry oxygen very well, and that's why it's called anemia. Okay? So these are abnormal blood cells. Individuals who are HbA, are susceptible to malaria. So in an area where malaria is endemic, if they get bit by a mosquito and they get malaria, they're going to get really sick and they might even die. Okay. If you have an individual who's homozygous for HbS, the abnormal, they're going to have sickle cell anemia. And they're going to have blood cells that look like this. Okay? And then if they're HbA, HbS, they're going to have a combination of a normal and abnormal. And those individuals, they won't carry oxygen quite as well as the normal, but they'll be protected from sickle cell anemia. I mean from a malaria, rather. Okay? So it means that they're heterozygous, and in this case, the individual with that in an environment with malaria has the highest fitness. If you're not in an area where there's malaria, the highest fitness is normal hemoglobin. So you can see how in different environments fitness changes depending on the alleles that you actually have. So when you have two different alleles like this, HBA and HBS, this is called heterozygous. So hetero, so here we are with hetero and homozygous that you referred to before, similar. So homozygous means the same alleles. Heterozygous means that you have two different alleles. So in this case, when you have two alleles, you can have three possible genotypes. You can have HBA, HBA, HBS, HBA, or HBA, HBS, and then HBS, HBS. Okay? So here's our... Okay. And we have a phenotype. So the genotype is what alleles the individual has. Remember, they have two of each. They can be the same allele or different. They're diploid organisms, right? The phenotype is how those alleles are expressed. Now, this isn't exactly right, but for now we're just going to say it is. We're going to say that HBA, HBA, and HBA, HBS have normal red blood cells. In other words, HBA is dominant. Individuals who have sickle cell anemia, they're HBS, HBS, they're homozygous HBS, they're recessive. You can only get that expressed if you have two S alleles instead of just one. Okay? So that's what dominance means. If you have a dominant versus recessive, an individual who has a dominant allele, he can have one allele or two, and they're going to express the same phenotype. HBA and HBA, or HBA and HBS, they have the same phenotype. And then the recessive is HBS. You need two copies of the recessive allele to express that. Okay. So, genotype, alleles that you have, phenotype, the traits that you have, including anatomy, physiology, behavior, development. So, in this case, the heterozygote HBA, HBS does not show the disease. In other words, the disease state is sickle cell anemia. Individuals who have this, their fitness is very low, if anything at all. Individuals who just have one copy, they're heterozygotes, HBA, HBS. They're carriers of the recessive allele. In other words, they can donate that allele to offspring. In fact, if they're HBA, HBS, half their offspring, they'll donate the HBS allele, and half the time they'll donate the HBA allele. Okay? Depending on the environment they're in, if you get HBA, and the father's HBA, and you're in an area where there's no malaria, the best fitness is normal alleles, HBA, HBA. If you're in an area where there's malaria, the highest fitness is HBA, HBS, because they won't be infected by the malaria parasite. Okay, so instead of HBA, HBS, we'll just say A1 and A2. It's two alleles. You have two homozygote genotypes. If A1 is dominant to allele A2, then the dominant phenotype is expressed in both the homozygous and heterozygous genotype. So A1, it's going to, of course, express a certain phenotype, which is a dominant phenotype. A1, A2 is heterozygous, but it's still going to express the dominant phenotype. It's only the recessive, A2, A2, that you'll get the recessive phenotype only if you have two copies of A2, A2. Anybody be able to do this with their tongue? Anybody in here? No one? There's got to be at least one. Okay. Oh, you are. Oh, great. Okay. Finally, we have a beneficial mutation. The rest of us are losers. Count your lucky stars. Anyway, tongue rolling is a dominant trait. All you need, now each of these things, all you need is one gene. It's monogenic. That means you have different alleles, but it's only one gene. So if you have one of those alleles, you'll be able to roll your tongue. You don't need two copies. If you have two copies, you will do it too, right? This right here, this is an earlobe, right? Well, my earlobe is detached, kind of like my personality. And so it's dominant as well. Now, there's also cases where there's recessive traits. In this case, you need two of the same alleles for this trait to be expressed. Cleft chin. You need two copies of the cleft chin allele for it to be expressed. If you have a normal and a cleft chin allele, that it'll still, you won't have a cleft chin. Hitchhiker's thumb, the same sort of thing. I think I got one, yeah, okay. So these are traits that are affected only by one gene. Okay? There might be many alleles, but it's only one gene, and in an individual who's diploid, they'll have possibly the same allele both times, or one and the other, and then it's heterozygous. Okay. We're back to hemoglobin again. Remember, we've got homologous pairs of chromosomes as in a diploid. And when you produce a gamete, you'll either produce an HBA or an HBS, okay, if you're heterozygous. Now, if you remember, we did this before with STRs. There's the same sort of thing going on in terms of the way alleles are inherited. Since mom and dad are diploid, they each have 2 alleles, and the siblings have 2 alleles, maybe the same, maybe different. The 4 possible outcomes, of course, are this, and this, and this, and this, and then this, and this, and, oops, not that, hold on. And those are the 4 possible outcomes. Okay. Now, you can figure out what genotype and phenotype an individual are if you know what their parents are. So let's say we have say this is the man, this is the woman. Heterozygotes. So they either have an HBA and they also have an HBS. So I'll just say up here, I'll put A or S. And for the females, same thing, A or S. So these are all HB before them. So in terms of determining, you want to know what % probability that your child will have sickle cell anemia. In other words, getting 2 copies of S. You take an A and an A. This is normal hemoglobin. You have a heterozygote, AS. And here you also have an SA. Those are heterozygotes. And then you have SS. So the probability is 25%. If you have 2 heterozygotes as parents, there's a 25% chance that an offspring will have sickle cell anemia. There's a 50% chance that they'll be resistant to malaria and will be carriers. In other words, they can donate that to future offspring. And then there's a 25% chance that you'll have 2 normal hemoglobin individuals. You'll have 25% chance for a normal hemoglobin individual. So I might give you, a bunch of different, I might say, here's a homozygous man and a heterozygous woman, what percentage will have, will be a carrier? Would be 50% actually. But we'll go through more of these as we go along. There's something kind of interesting. You know about bicyclists and runners who, they use something called EPO. It's against the law, but what it does is it makes you produce more red blood cells. So your blood can carry more oxygen. When you go up in altitude, that's what happens. Over a couple of days, your body goes into overdrive and starts producing extra red blood cells. The only downside is that your blood becomes more viscous, and there's more likely that you'll end up with a clot, and because of that, perhaps a heart attack or a stroke or something like that. So, like people in the Andes, they have normal hemoglobin, and they probably have higher incidence of blood clots and things like that. Well, Tibetans aren't like that. Tibetans actually have a different form of hemoglobin, that there was mutations that evolved, and their hemoglobin, instead of having to have more red blood cells, they don't need more red blood cells. They'll carry the same amount of oxygen. And so they have a real advantage. And so if you go to Tibet and the Sherpas and all that, they all carry this gene. But it's a gene with a lot of different mutations. And so it was thought, it was just a matter of natural selection. This population developed a mutation, spread, all that kind of stuff. Well, they sequenced a Denisovan, which was an archaic human that humans bred with and produced hybrids. Denisovans, they came before us. It turns out that mutation actually arose with Denisovans because they actually recovered DNA from them. They sequenced it. There was this area that controlled respiration, cellular respiration, and oxygen and hemoglobin. And they're the ones that actually passed down that adaptive mutation to offspring, hybrid offspring with humans. So it turns out that the Tibetans didn't, on their own, someone developed that mutation. It was actually the Denisovans. There was hybrid sexual reproduction, like one male and one female. And some of their offspring ended up with the mutation. So the Denisovans are extinct now, but the Tibetans are still around, and since it's such a beneficial mutation, everyone in Tibet actually has that mutation. They have because babies having that mutation as well are much more likely to survive till term than if they don't have that. So I just thought I'd pass that on. Kind of cool stuff. That's it.
Thank you.